Okay, thanks for the introduction, Philip. I'd first like to start by thanking the conference organisers for the invitation to come here today and tell you a little bit about our thinking in this area and fill you in on a bit of the work that we've been doing over the years. It's a pleasure to have this opportunity. So today I'll take you through the things that I've got up here. It's going to be something of a sort of historic overview to start with, to sort of explain to you the sort of simplicity, the naivety of our thinking when we first got into this area and how it's had to evolve as we've come to understand more and more about the microbiota. So I'll talk to you initially about our interest in trying to identify performance-associated microbial signatures, the sort of bacteria that are involved in giving us a good performance in birds, the use of probiotics, how they're used at the moment, and then I'll sort of step back a bit again and talk a bit more fundamentally about functions within the gut and the microbial interactions, because I think we have to think a bit more deeply about the microbiota and how it varies along the gut, and it's not just a single sort of uniform tube, you know, we have to think of it in a slightly more sophisticated way than that. I'm also interested to just briefly mention something about the origins of the gut microbiota. Where does it come from? How does it get established? And what does that mean in terms of things like the innate sort of variability of the microbiota? And that helps us to start to understand the sort of intervention points where we can go in and try and change that microbiota or repair it, or, you know, how we can intervene to try and get the most out of our birds. So, you know, why is the gut microbiota a good target to look at to try and improve health and productivity? Philip's obviously gone into a little bit of this in his introduction to the meeting, but we have to think of the microbiota as really an important, it's almost an extra organ that we have in our bodies. So it provides us very specific functions that are essential to the health and well-being of all animals, essentially. And the gut microbiota, obviously, because of its location and what it does, has really big impacts on gut health, and we recognise that more and more, both in the animal health industries, but also in the human health area. And we do work in both areas, both in poultry and in animal health, and each area of work informs the other area of work. A lot of the sort of fundamental stuff really has been done in animal health, and it's now being applied into human health. So it's one way in which, you know, we've really helped the human health practitioners. And of course, gut health is so important to the overall health and productivity of birds. You know, the things that we really need to focus on as an industry to get the most out of the resources that we put into producing the birds. And the sort of underlying sort of concept here is that as animals, as humans, you know, we're not just our own human cells walking around, but essentially we're a meta-organism. It's essential that we have our gut microbiota, and it's the unity of our gut microbiota and our own sort of biological potential that produces the organisms that we have out there functioning in the world. So there is this concept around of the meta-organism that captures that idea. So just to remind people, you know, I wasn't sure exactly how familiar everyone would be with sort of microbiota research, so I just put this up to remind people of what the microbiota is. It's all of the microbes in a particular environment, and we'll be focusing almost exclusively, I think, on bacteria here, but the microbiota does include other things such as the Archaebacteria and the Protists, fungi, viruses, so they can all have something of a role, but we'll be concentrating mainly on bacteria because they have the largest functional role, and it's lucky for us that they're also the ones that are perhaps easier to characterise using the sort of sequencing technology that Philip talked about. Now, I go back a long way further than Philip, so when he started getting 100,000 bases in a run or something might be good. When I was doing it, 100 a day, if you could average that, would have been fantastic. I spent a whole year of a postdoc to sequence just a few thousand bases. So the technology has come a long, long way, and I'll mention that again in a minute or two. To jump right in and to just list some of the many important functions that that host microbiota interaction drives. Principally, one of the very important things that the microbiota does is to release nutrients that the host can use. But of course, the microbiota also competes to use nutrients. So there's this balance between freeing up nutrients that the host can use, but also retaining nutrients within the bacteria themselves. Really importantly, the host microbiota are essential if we're to digest resistant carbohydrates that we don't actually have the enzymes to digest ourselves. So it's essential to get that the biochemical potential of the microbiota to be able to get the full energy from those resistant carbs, and really importantly too, to produce short-chain fatty acids from those resistant carbohydrates. And the short-chain fatty acids is a recurring theme that I'll come back to a few times during the talk. Both in animals, but really, really importantly in humans, the microbiota is essential to get proper immune development. Both in terms of actually developing the immune system so it can see foreign invaders, but perhaps even more importantly, to train the immune system to not see common food components, common gut bacteria as foreign, and so to avoid unnecessary inflammatory responses, because inflammatory responses are a really big drain on the energy reserves of birds. So the way the microbiota sort of orchestrates the immune response of the host is really a critical issue. The microbiota also are really important in establishing the gut structure and integrity, the amount of mucin that's produced, the size of the villus, providing energy for cell proliferation in the gut. It affects the vascularization of the gut, the musculature of the gut, the motility of the gut. So the interaction of the microbiota with the host gut has all these effects that are really very central to us getting the most out of our birds. The microbiota in the gut can also produce toxins, and it can detoxify toxins that come in the food too. So there's sort of two aspects to that. Another really important aspect of the microbiota there are that they produce micronutrients that we can't produce ourselves. So they produce some of these things like vitamin K and folate that I've got listed there that we can't do without. We can't make them ourselves. They're all also important in recycling of bile acids, and they feed into endocrine hormone production. I'll touch on that a little bit later as well. And of course, we all know they've got a central role also in the sort of environment that's set up in the gut and whether that can be invaded by pathogens. So an important application of sort of microbiota technology at the moment is trying to exclude those pathogens from the food chain. So the next couple of slides are sort of quite general in terms of the sort of functions that the microbiota have, just reiterating some of the things I've said and expanding on them. So this is from mainly a human point of view, but it all applies to chickens as well. So I've sort of broken it down into four main areas: the sort of digestion, nutrition, the actual function of the gut, the immune system, and an area that's come up a lot in the last few years, particularly in human health, is this gut-brain axis. There's a very strong connection between what's going on in the gut and what's happening in the brain. That's through endocrine activity, but also through direct neural activity as well, linking the gut and the brain. So when these things are all going well, when there's a good balance in microbiota, it's functioning nicely with the host, the functions that I've sort of listed up here are working well and things are going okay. But when things are out of balance, you get dysfunction. And, you know, in humans that can lead to obesity, and of course in animals we're more worried about poor growth. In humans, food allergies, in gut function. In humans it might be irritable bowel syndrome and related syndromes. Clostridium difficile is really important in humans. In poultry, you know, we could replace this with Clostridium perfringens as a problem when things are going wrong. And in the immune system, you can see inappropriate inflammatory responses in things like asthma and diabetes. And really interesting in humans, there's a fair bit of work now going on to produce probiotics for some psychiatric diseases and also things like autism. So there's great potential in this area. But getting back to birds more specifically, I'll now tell you a little bit about the sort of work we've been doing and really start with the incredibly naive, sort of simplistic view we had of what we were going to do when we got into this area, I don't know, about 8 years ago, something like that. So we're going to go in, identify the bacteria that are more abundant in the high-performance birds, pull them out of birds and use them as probiotics. I mean, it's really easy, no problem at all with that. So we started our research, and our goals were really to catalogue and define the composition of the gut microbiota, to try and understand it, understand how it's established and the development of it over time, and then to drill down into that and understand the role of specific members of the microbiota and the effects that they can have on the health and productivity of birds. And then finally, of course, is to use that knowledge to try and develop ways to modify the microbiota to improve health and productivity outcomes. And we were specifically interested in broiler chickens at this stage. So to start with, we took a somewhat different response, different approach to the way most other people were tackling this work. So the way, productivity trials have generally been done is to put in a few variables into the system. So you might put a couple of different feeds or a feed with an additive in there, and you might have a couple of different environments or different lighting conditions or something like that. So you have a series of variables that you look at. And we thought this was sort of getting a bit too complicated for our sort of simplistic way of thinking about this. So we decided not to go down that usual path of trying to design these checkerboard experiments. And instead we went with just a single group of animals. So we tried to get rid of all variables that we could. So they all had the same feed, the same environment, they're from the same stock. And then we were interested in looking at the natural variation within that group to see if that could tell us anything about the relationship between what's in their gut and the productivity of the birds. So, doesn't matter what performance variable that you measure, you're going to come up with this sort of bell-shaped curve here. Here I've got body weight, but it could be FCR, it could be apparent metabolizable energy, it could be time to market, anything you put, it's going to have this sort of bell curve. And that means that on that curve you've got sort of low performance birds and high performance birds. So what we were wanting to do is get rid of all the other variables so that it was just down to the variability of the performance and see if we could link that with the structure of the microbiota. So when we did that, you analyse the microbiota within a group of birds, individual birds this is, and you get a particular structure of the microbiota, just to sort of fill people in. This is just a graph showing that the composition from bird to bird. So each column is one bird, and the different colours are different aspects of the microbiota and different proportions there. So when you get that data, you can then do some statistical juggling to see how closely related one particular microbiota structure is to the next. And when you do that, you can then map those figures onto a tree of relationships. So the closer they are in one of these trees, then the closer the samples are in the structure of the microbiota. And there's really only one sort of take-home message from this, and that is that the high-performance birds and the low-performance birds tended to group together. So there are consistent differences between the microbiota in the high-performance birds and the low-performance birds. You know, there are exceptions, there are outliers, but by and large, they do separate into different groups. So this tells us that there are things driving the differences in microbiota between the high and low-performance birds. So microbiota clearly is having an effect here. However, this is when our sort of simplistic ideas started to need some modification. So, you know, we're not working in industry, we're working in science, we're trying to get stuff published, we're trying to understand the sort of rational scientific basis of these things. And one of the sort of fundamental things we have to do as scientists is to show how reproducible the work is. So that was one experiment, but what happens if you do it over and over again? Do you get the same result each time? So with support from our industry body in Australia, we went ahead and replicated this trial. And the good news was that in each experiment, we did find bacterial types whose abundance was correlated with performance. So there was always bacteria that seemed to be driving that difference in performance. The bad news was that the performance in each trial was associated with different bacteria. So each time you do the experiment, you certainly get bacteria that are driving performance, but generally it's a different sort of bacteria from the previous trial you did. There's some overlap in the groups of bacteria and certainly the types of bacteria that are related to performance, but they do vary from trial to trial. So that has a number of consequences in the way you may think about going forward with this. But this is just to sort of visualise some of the data that we were pulling out of this. So when we repeated the trial, this is a different way of looking at the data. So the larger coloured dots there represent the animals. So that's a particular animal, and the tiny dots are what we call OTUs, operational taxonomic units, which are essentially like species of bacteria. So they're a particular sort of bacteria at about the species level. So you can see there's lots of little black dots. That's because there's lots of different species of bacteria in all these birds. And within these two trials, most of the black dots fall between the bigger dots that represent the birds. So that shows that they're shared between those groups of birds. But there's some OTUs, some sorts of bacteria that are only in one trial or the other. These ones up here and down here. But by and large, they were fairly similar. But when we put the third trial in, that's when things got really hairy, really different. And in this third trial, and these birds are performing just as well. I mean, in all cases, these birds are really high-performance birds. So even when I say high and low performance within a group, they're still all, you know, really pretty good performing birds. But in this third trial, you can see that there are a huge number of bacteria up here that are only connected to third trial and have no representation at all in trials 1 and 2. So what do you do with that sort of data? I mean, how do you sort of deal with it, and what does it mean in terms of development? So this is just, you know, I talked about sort of statistical juggling to try and get the relatedness of bacteria within one bird and compare it to the next. But I put this up to show just how different these trials are. So there's 3 trials represented here with about I think it's 30 birds or so from each trial. But you don't have to do any statistics, you don't have to do any analysis to clearly see that there's 3 different sorts of microbiota here. I think you can clearly see that without any statistical juggling. So that's the sort of variability that we have to deal with if we're going to do something with the microbiota of these birds. So just to reiterate, each trial there are OTUs correlated with performance. The OTUs, these sorts of bacteria, they tend to be different in each trial. And we think that this complicates how you might go about manipulating the microbiota. So if you pull out a bacteria that's important to the performance in one of those trials, will that actually work in the other group of birds which have a different underlying microbiota? This is really fundamental, I think, to some of the variability that we see perhaps with some of the probiotic products and even, prebiotics that we use out there. Sometimes when we use them, we get really good responses because they're going into a microbiota that's appropriate and they can work within that. But sometimes we don't see such good responses, and that may be because we're trying to use them in a background microbiota that really isn't particularly suitable for them. And, for us as scientists, we think this presents us with opportunities to really try and develop new products, principally probiotics, in a fundamentally new and different way that really takes into account this microbiota variation, which is a fundamental finding in poultry now. So with the work we've done on looking at performance and the structure of microbiota, as Philip said, there's an awful lot we've learned over the years, but there's a lot more that we don't know still. And I just put up here a few themes that come out in both our work and the work of many other groups around the world in terms of what actually is linked to a high-performance microbiota. One of the things is that the overall structure of the microbiota tends to be more complex. When you've got dysbiosis, something's going wrong, you tend to have less complex microbiota. It's simpler, that's dominated by fewer species. So high complexity seems to be good. A lot of people will monitor OTUs, lactic acid bacteria, as an indicator of whether the microbiota is healthy or not. And, we'd agree with that to some extent, although we'd caution that, we've certainly found Lactobacillus strains that seem to be associated with poor performance as well. So it does work both ways. But generally OTUs, related to the different groups of lactic acid bacteria are thought of as good, but be cautious. Low Proteobacteria is usually a good thing. The Proteobacteria include a number of important pathogens, E. coli, Salmonella, and others. And so generally we find that a high-performance microbiota is lower in those groups of bacteria. And then we get on to the presence of other more recently identified bacteria that seem to be quite beneficial. I've got some names up there, you can read them. The Archaebacteria Prusnitzii seem to be, has a fundamental role in the development of the immune system. Akkermansia eosinophilia is involved in production of mucin, and use of mucin in the gut seems to be involved in the repair of the gut. Quite important. And then there are a group of bacteria that seem to be really important in short-chain fatty acid production, and in particular butyrate production, which is an important energy source for the gut. And so there's a group of bacteria that are now being characterised, including by Philip's group, including several different sorts of Clostridia. The important thing to note, though, is that there isn't one, at the moment, we can't define one ideal microbiota. There seem to be many different routes that you can take to get to the end result of a good functional microbiota. And so, it may vary too between the sorts of diets that you're using. So, a production system that's using mainly a wheat-based diet may have different underlying microbiota structure to one where corn is the main energy source. So, just to put up a little bit of real data here that this is illustrating the complexity issue that I talked about, that it seems that complexity is good in the microbiota, and this is an experiment we published last year, I think, in which we're looking at the development of the microbiota over time. So from day 1 out to day 23, and it's at about day 20, 23, something like that, that we seem to get stabilisation of both the complexity and the structure of the microbiota. So here you can see that the microbiota is, so each of these is a richness level, so the higher the richness value on the axis, the more diverse the microbiota is. And each of those spots is a bird and the richness of its microbiota. So you can see that within any one group at any one time, there's a very significant variation from bird to bird, but the trend is to greater complexity. And then you can drill down into that data, and you can look at very specific members of the microbiota. So here I mentioned Ficalibacterium as important in immune development, and here in this experiment we see a general trend towards an increase in the levels of Ficalibacterium over time, whereas other things, here the Enterobacteria, are reducing, which we think is probably a good thing. So just to harp on the complexity of the microbiota, these are old figures really. I mean, these were generated probably before the sort of modern genomic era that we're working in now, but the thought was that there's something like 600 to 1,000 different bacterial types within the gut of chickens. And as Philip mentioned also, the majority of those can't be cultured. We can't pull them out and do anything with, but we can analyse them. We can understand what's there. It's just that it's very difficult to do anything with them at the moment. But there is a lot of new technology being thrown at that as well to try and allow us to access the potential of all the bacteria that are in the gut. Now, I say there's 600 to maybe 1,000 distinct bacterial types, but even within each of those types there's going to be lots of strain variation from strain to strain. So, you could have a particular bacillus, say, Bacillus subtilis, which all look very similar, but they can have just one gene different. They could be expressing an antimicrobial protein that can kill Clostridium perfringens, and it can make a fundamental difference in how those two organisms work. So even though there's only a single gene difference amongst the whole genome of those organisms, they could be quite different in their actual effect on the gut. And as I've shown, the microbiota certainly varies flock to flock, but what I didn't sort of really point out clearly was that there's a lot of variation within a flock too. So just bird to bird within a flock, there's a lot of variation in the structure of the microbiota. And importantly, even though those microbiotas varied a lot, they're all high-performing birds. The other thing to keep in mind too, when we're thinking about how to use these and using probiotics, whether it's single strain, multi-strain, or trying to pull apart the microbiota to understand their role in performance, you have to understand that these things aren't working in isolation. They're working as a community, so there's a whole network of functions. So some bacteria will produce a metabolite which other bacteria can then use and produce other metabolites. So, one bacteria may be good at producing a particular metabolite, butyrate or something, but it's no good if it doesn't have the feeder system going into it. So, as Philip said, we've got an awful lot to learn simply because of the complexity of this. And so, these are the sort of questions that are keeping us awake at night. the first question here, well, I think I've clearly shown that there are different microbiotas and they can have different health and productivity outcomes. But we're interested to try and understand why the microbiota is so variable. And can we do anything to actually control that variability? Is just the control and standardization of the microbiota a worthwhile goal in itself? So, as the trials that I've talked about, we did everything we could to try and control the variation. So we had the same batch of feed, the same water, the same environment, the same people handling them. They were going into the same rooms, they come from the same genetics. So with all that controlled, we still got variation, and that's on top of the fact that, of course, chickens are coprophagic, so they're eating their own poo all the time, so you've got this recycling of the microbiota in the gut, and it's being taken up by the other birds within the flock. So even with all that going on, there's still a lot of variation within a flock, and why is that? So we and others, it's certainly not our original idea, hypothesised that the hygiene levels in modern commercial chicken hatcheries is really the fundamental cause for a lot of the underlying variation in the microbiota. And that's because these days, newly hatched chicks are not exposed to the natural microflora of birds. So they're not, the eggs aren't laid in a nest, the hens don't sit on the chicks as they're the eggs and the chicks as they're brooding them. But instead, the birds are coming out of these clean environments that, we keep clean for very good reasons, to try and control salmonella and Campylobacter. But that means that the birds aren't exposed to natural bird microbiota. They have no exposure at all to that. So the only exposure they have to microbiota, the only place that they can establish their own microbiota, is from all those random meetings with other bugs that they have. So from the hatching environment, the people going in there and handling them, the first feed they see, the bedding they're put in, the receiving environment. This is the source of the microbiota that's establishing in the guts of our birds. It's not natural bird microbiota. And because of that, there's a great variation, in what they're picking up, whether one bird gets it mainly from a handler who's picked them up and put them in the transport box, or one bird's getting most of its first dose of microbiota from the first feed it's seeing. And it's really a sort of random, stochastic process. But the other important aspect of this, we believe, and it's difficult to get evidence for it at the moment, but we believe that the founding bacteria, so the first bacteria that go in, can have fundamental effects over the life of the bird. So what gets in there early actually influences how it develops after that. So what we're seeing out there and what we're having to deal with is microbiota variability that we believe is principally caused by this underlying process. So the microbiota in birds is really fundamentally, the acquisition of it is fundamentally different to mammals, because, of course, in mammals, with vaginal births, that's the first source of microbiota generally. So mammals generally are acquiring the host, the animal's microbiota right from the get-go. Whereas in birds, as I say, modern production, it's a completely random process, unrelated to the adult birds. At the sort of overall phylum level, the sort of bacteria that are in there, though, between mammals and birds, it's really not that different. It's driven by the groups of bacteria we call the Firmicutes, the Bacteroidetes. There's Proteobacteria, which we think, it's nice if they're fairly low, and a number of other sort of lower abundance types of bacteria that are in there. And in birds, as far as we can tell, certainly in modern production, the microbiota is pretty much established and fixed after sort of 2 to 3 weeks, and then it doesn't change an awful lot over that the rest of the time unless something happens, unless something's going wrong with the birds. And it's really when things are going wrong that we should be thinking about using interventions. And that's quite different to certainly humans, where the microbiota tends to develop over a few years. So it's a much longer process. I've talked about the diversity increasing over time. And the highest diversity in the gut of the birds is really found in the caecum. The caecum is this sort of fermentation powerhouse that's helping the bird acquire nutrients from resistant starches. As a microbiologist, I find the upper gastrointestinal tract, so the sort of duodenum, jejunum, even the upper ileum, to be pretty bloody boring, actually, because it's usually completely dominated by Lactobacillus, so it doesn't give us a lot of sort of really interesting things to pursue there, but they're really important, obviously, in the whole digestive process for birds. So with this sort of background information that we're sort of building up as we go along, we think there are sort of some really interesting new opportunities to develop products in this area. In particular, microbiota establishment products. So this is going back to some of the early work from the 70s with NERMI and the use of very crude cecal extracts to try and colonize birds and make them resistant to infection with salmonella. We think that actually looking at that early establishment is really an important opportunity for products. So rather than dosing probiotics throughout the life of the birds, can we go in at hatch and really help the birds establish a good microbiota, put them on the path for a successful meta-organism development over the course of their life. The other opportunity is, given the variability that we see in performance, can we use probiotics not necessarily to increase the productivity of the whole flock, but to really narrow the variation that we see within a flock? Can we make the performance more consistent, predictable? And of course, where a lot of probiotics are used today, trying to fight off pathogens such as Clostridium perfringens, I think there's still a lot of work that we can do there to help that process. In the time remaining, I just wanted to step back a bit now and go back to the gastrointestinal tract and think about the microbiota and what we want to do with the microbiota really in the context of the whole gut rather than just talking in general terms as I have up to now. So this is the real gut, but I find it a bit difficult to annotate that and do things with it, so I've simplified it with this cartoon, which I'll use to talk about, firstly, the functions of the gut. So each part of the gut obviously has very specific functions, and it'll only happen in that part of the gut, not in other parts of the gut. So we have to have that in mind when we're trying to manipulate the microbiota. But as well as those functional differences, there are also physiological gradients that are happening across the chicken gut, whether it's pH, which I've shown here in cartoon form, the level of oxygen in the gut, which decreases as you go further down the gut, and in the caecum it's essentially free of oxygen. The nutrient content of the gut content that's going down there, whether it's protein or carbohydrate, obviously nutrients are being extracted as we go further down, but things like resistant carbs are increasing in the percentage that's there. So all these things are happening in the gut, and on top of that there are gradients in the bacteria there. There's gradients in the actual absolute numbers of bacteria that are there, which peaks in the ceca. There's maybe 10 to the 11, 10 to the 12 bacteria per gram in the ceca. It's basically just a bacterial paste. Whereas Lactobacillus, they're more abundant as a percentage of the microbiota in the small intestine and reduce as you go further down the gut. But then there are other really important bacteria, like some of the butyrate producers, which are often strict anaerobes, which increase as you go further along the gut. So all these things have consequences in what sort of bacteria you might use. Probably don't have time to go through that again. So along with those functional regionalisation throughout the gut, the physiological gradients, the microbial gradients, there are particular interactions between the microbiota and the gut that are important, and they are regional things. And I've just listed them, some of them up here, and I won't go through them all, I don't really have time, but you have to have the right bacteria in the right part of the gut. So, the one at the end there, bile deconjugation, I mean, that's an important process to be able to recycle bile and reuse it and all. But you can't have that happening in the wrong part of the gut. Then you won't get proper fat digestion, and you won't get proper growth of the birds. And also, the bacteria that are involved in development of the immune system, they have to be in contact, obviously, with the immune cells that they act on. Can't be in some other part of the gut, It's no use having some of the butyrate producers in the upper part of the gut if you haven't had any pre-digestion of the complex carbohydrates to feed them. So now I'll just touch on how we might go about manipulating the microbiota, and there are obviously two ways to do that. One is to do it directly by introduction of probiotic bacteria. The other way is to do it indirectly by the use of prebiotics, so things that actually feed bacteria and try and encourage the growth of the bacteria that you're interested in helping. And there are obviously a lot of commercial products already out there, so here's a list of some of them that you can find out there on the web. I'm not claiming that this is an exhaustive list in any way, but it just is there to make the point that there are a lot of products out there that are already using our knowledge of the microbiota to try and give some commercial advantage to farmers. But I'm interested in sort of actually looking at some of the claims of these products, and I've just pulled out a few of these just to make a couple of little points. So these are product claims straight from the websites of some of these products. So inhibition of pathogens, e.g. Clostridium perfringens, is obviously a big one. A number of products are directed at Clostridium perfringens, and they clearly have activity against Clostridium perfringens and work well. So that's fantastic if you've got a necrotic enteritis problem in your flock. But what's the consequence of using those products if you don't have a necrotic enteritis problem? I mean, if they're killing Clostridium perfringens, are they going to be killing other Clostridia that might be involved in producing butyrate, an energy source for the gut? So I think you have to be judicious. You can't throw these things around in all situations. The second one, the claim was much broader. So this product is inhibiting Salmonella and Campylobacter and E. coli, and you could think how they could all potentially be affected by a similar thing, but it's also affecting spirochetes and most other pathogenic bacteria. Well, sorry, as a scientist, I'm not sure what sort of magic bullet can only target pathogenic bacteria and doesn't hit some of the good bacteria that are in there. I don't know the mechanism for that, and it would be great to support these product claims if there was some scientific basis for these sorts of statements. And, I guess the last and most general one up there is, improving the balance of beneficial microflora. That's great, and, it's the sort of thing you see on advertisements for human probiotics and all. It makes you feel wonderful and all the rest. But, what does it really mean? I mean, how is it changing the microbiota? And under what circumstances will it work? So these are the sort of questions that I'm interested in as a scientist and sort of trying to address that and understand that. I'm certainly not trying to dismiss these at all. I think they genuinely do have some really useful and beneficial functions, but we need to understand what they are and how they work if we're to try and develop a second generation of products that are perhaps more effective. given the time sort of running out, I'll just jump to the bottom point here. And it's my personal view, and I'm sure there may be many out there that'll disagree with me, but the evidence that I see is that at the moment probiotics really have good application when something's going a little bit wrong with the flock, when you've got dysbiosis or you've got a particular disease issue, necrotic enteritis, whatever it might be. So I think these products seem to be quite good at rescuing performance, but in our hands we're really struggling to see much effect of these products when you put them into a high performance flock. So if we put these products into a high performance flock on a non-antibiotic, non-coccidiostat feed that are, got high biosecurity, are performing really well, we really struggle to see much improvement when we use the current range of products. And I don't think that's surprising. I mean, the whole point of these products is to try and improve the gut microbiota, and if the gut microbiota is already fine-tuned and working well, why would you think it could do more? So I think we shouldn't look to them as magic bullets to improve things when they're going well. So I've got a few examples here of some attempts we've made to modify the gut microbiota. In the first one here, we use just crude cecal content. The cecal content, so this is just showing the structure of birds that we isolated the cecal content from. Here and here, we took two groups of birds that had very different microbiotas. So this one over here has a very high level of Bacteroidetes, and we did that just so we could distinguish them apart. So we took those pooled cecal samples from these birds and put it into naive birds at hatch. And what we actually found was that the naive birds at hatch sort of picked up their own microbiota from all those random things that we talked about. And we couldn't actually drive the microbiota of the birds by adding this in the way we did in this particular experiment. So this is the FCR results for what we put into the birds. So this was our poor FCR group, this was a good FCR group, but the birds we put it into all had good FCRs. Again, it's this problem that we've got all the time. The people that we do these animal experiments with, they pride themselves on performing better than the industry standards, and they do all the time, and it makes it very difficult for us to get meaningful results out. But then we went from using a sort of very crude total cecal extract to using a mixture of 6 specific cultured LABs that we pulled out for various reasons, well, very specific reasons. And when we put those into birds, we were able to find that we were able to get increase in weight gain, statistically significant over the 28 days of the trial. And this is another way of showing the structure of the microbiota of these birds. So this is, the closer they are, the more similar each of these birds is in microbiota structure. And the only point I really want to make here is that whether it was the control birds that didn't have any of these LABs deliberately put into them, or our group of LABs that we put in, they do end up with very different bacterial microbiotas over and above what we've put in there. I mean, it's driving changes in the rest of the microbiota. We've also done work on feed supplements. So here we're looking at biochar, bentonite and zeolite, sort of biochar you'll know about, and bentonite and zeolite are sort of clays. And when we do that, we get different microbiota structures from each of those birds, and perhaps more interestingly, we're finding that compared to the controls, each of these is reducing the level of Proteobacteria, and specifically Campylobacter in these birds. So that's something else that we're pursuing as a way to manipulate the microbiota. So not just going down the probiotic route. Specifically with zeolite here, we're showing a reduction in different sorts of Proteobacteria as well, which is an interesting property. So one of the things that always frustrates me with the current probiotics is the need to continually dose them. Now, I understand that that's a fantastic product for the producers, the people who are selling them, but to me, if you've got a really good bacteria that's doing something in the gut, and if you select it and choose it in the right way, you should be able to give that bacteria to the birds and that's it, they're set for life. So that's my recent vision of what we're trying to do. So we're trying to move away from these continually dosed probiotics to probiotics that you can give either at hatch or even later on in life. And they can actually get in there and establish themselves and produce their positive effects. So most of the probiotics, as I understand it, and I'm not privy to all the commercial development that goes on within companies, but my understanding from the literature is that most of the current range of probiotics have initially been identified by applying in vitro tests to try and identify those that would have good properties in terms of survival in the gut. So it might be resistance to bile or the acid conditions of the gut. It might be adhesions to the gut cells, so things that might be able to stick around in the gut and stay there. However, this is likely to be fairly controversial, but if you actually look at the literature, there's virtually no controlled trials there where people have pulled out things that performed well in these in vitro assays and things that performed poorly and actually compared them. All they ever do is get positive strains from these in vitro assays and put them into birds. There's never the negative control. There's never any formal scientific proof that these methods are actually telling us anything. And we've got a fair bit of data at the moment that shows, certainly in our hands, that we've got strains that perform really well in these in vitro tests that don't perform well when you put them into birds, and vice versa as well. So we've got strains that don't seem to be particularly bile or acid resistant, yet they perform quite well when you put them into birds. They can persist in there and they seem to have some positive effect on productivity. So we think there's a new way to go about developing probiotic strains. And that's really to put them through animals to start with. So we're coming up with high throughput screening methods that allow us to actually screen many strains of bacteria directly in birds to see those ones that can persist and stick around and do something in birds rather than just disappear. And the surprising finding is that even when you go to birds, a lot of the probiotics that we're using in the industry at the moment aren't even derived from chickens. They might be derived from food sources or other animals or environmental sources. But even when you go to chickens, you pull and culture bacteria out of their guts and then put them back into birds. There's only a very small minority of those chicken bacteria that can reliably get back in and re-establish under variable conditions. So, you can vary the feed and they'll still be able to get back in there. So that's a sort of an interesting finding that I think is quite important, and it emphasises why there's this need to be able to come up with methods to identify those rare bacteria that have these surprisingly unusual properties. And so we're starting to use these methods to pull out bacteria, and where the targets that we've got to use these bacteria are all the same as the ones that we've talked about up to now, but our hope is that they're going to be more effective and perhaps most importantly more reliable, so they're going to be able to go into any sort of microbiota rather than just ones that are suited to that product. So that's where I'll sort of finish the talk really, but just leave you with a few more fundamental questions that have arisen as we've gone along and done our research. We're particularly interested in the origins of all the microbiota, particularly the strict anaerobes. I mean, where do they come from in the environment? I mean, some of them form spores, and that's a good indication of how they survive out there in the environment, but there are a lot of strict anaerobes that don't form spores. So how do they how do they transfer from, whether it's the human handlers, the feed or whatever, to the gut where, there's this very special environment that they can survive in. So we really don't understand that. I mean, it's a very fundamental question, but there's still an awful lot of work to do. And what are the relative importances of those different sources of microbiota that I've talked about? Is it the first feed that they go on, or is it contamination that might be on the eggs, or is it the people handling it? Is it just stuff circulating in the air? What are the most important sources for birds at the moment? And also, I mean, I've mentioned that we analysed microbiotas from different groups of birds that were all performing well, and they have quite different microbiotas. So how can that be? How can they deliver similar functional outcomes even though the structure of them is different? And what is it about that interaction between all the bacteria there and the host that really, without our intervention, can come up with this incredibly complex but different structure that works so reliably most of the time? It's an amazing sort of evolutionary feat that this can come together and work so well. And really, how do these changing networks, so when there's a few things that go in at the start and then they gradually get more complexity as they go on, how do they stabilise and stay the same after a while? So these are the sort of fundamental questions that we'd like to be able to address, although how you tackle them is not always clear at the moment. Lastly, I'd just like to acknowledge 4 of the really key people. There have been a lot more involved in the work, but these ones have been really important in the work that I've talked about, and the support of our industry body, the Poultry CRC, which has supported the work for many years. Thank you.